Okay guys, so in this video we are going to talk about the biosynthesis of phenylalanine. This is a very important molecule in shikimate pathway where we encounter this molecule in so many times in biosynthesis pathways, especially in shikimate pathway. Okay, so let's start the mechanism. We are starting with crosmate, or otherwise the crosmic acid. If you are wondering what is this or how we can synthesize this particular molecule, we have talked about in our previous video, so I'm going to put the link for that particular video in upper right corner. You can watch it if you need to know about more, like how this molecule came to the action for this particular molecule, for this particular biosynthesis pathway, right? Okay, then let's move on to our today's mechanism. The first step now, I just want you to listen very closely. Some rearrangements will be happen. The first rearrangement is, you can see here we have a double bond. This bond will be broken into here. Now you can see this carbon is unstable. This carbon is unstable. What happens? This bond will be broken back into here. Now this carbon will be unstable. Only option is to break one of these bonds. So one of these bonds will go and attack to here. This is the pathway. So what happens? You can see this bond will be broken. So you can see this bond has broken. Here double oxygen has formed a double bond with this particular carbon. It has formed a double bond with this particular carbon. So if I put a number 1 for this particular carbon here, you can see this is the number 1. You know that there's another carbon here too, right? This is a CH2 group, not the oxygen group. This is a CH2. I have told you this same thing in our previous video too. I hope you are not complicated with this CH2 group, right? Okay, this CH2 group is on this position. You can see this is the bond between this carbon and this carbon. This is the bond. I hope you got the rearrangement correctly. Now we have this molecule. We call it as prephenate. This is a prephenate anion. Now in the next step, you can see first step, like the two things will happen. First thing is that carbon dioxide will be removed. Carbon dioxide will be removed, and H2O, a water molecule, will be removed. So what happens will be, you know, there's a OH group in here. There are protons on our environment. This will attack here, you know, which makes a water molecule is a very good leaving group. So this one will be broken. That's how the H2O molecule will be removed. And the carbon dioxide, how will this carbon dioxide removed, right? You can see here. So let me, like, I cannot erase this, I guess. Okay, we were here, right? So let me draw this molecule again for just to explain it, right? Here you have this molecule. Here you have the molecule just like this. Here you have carbon and oxygen minus this is our molecule, right? Okay, now we have it like this. Now here you have O H. Now the proton has attacked here. So now you can see, like, this is a H2O group, which is a very good leaving group. So this bond will be broken. Why is that? Because there is resonance structure, like rearrangements can occur. This bond over here will be coming to here, and this bond will be broken into here. Because of that, this will come here, which will unstable this particular carbon, which resulting in breaking this particular bond and releasing water. So as I have told you in before, these are enzyme driven reactions, right? These are enzyme driven reactions. So that's why these are possible, right? Otherwise, we would wonder, like, how these things happen, right? What is the cause of this? Like, is there any nucleophilic steric hindrance? Likewise, like, no, like, these are enzyme driven reactions. That's why we are calling it biosynthesis pathways. So this is what happens in this particular reaction. So what the result is, you can obtain the result is this one. Here you have benzene ring, and our carboxylate anion. And here I hope you remember this molecule part. This is glutamate. This is glutamate. So transamination would happen here. A transamination would happen. This amine will be transited to this particular place. So here we have our phenylalanine and our other product. Basically this glutamate part here. I have Mr. Oxygen here. Like you can see, there's oxygen here. Right? I'm very sorry. There's oxygen here. So that's how the transamination works, right? The transamination work like amine group is going for the carbonyl part, and carbonyl, like this oxygen part, is going for this particular transamination. So you can see right here, the glutamate originally has the amine part. It's now currently on our phenylalanine, and now the oxygen which was present on our original molecule has been transferred into glutamate. So that now this is not the glutamate, this is our different molecule. So this is our main product. This is what our target is. So I hope you got the idea about this particular biosynthesis. I highly recommend you to watch our previous videos, which we have discussed about the cosmic acid, how we can biosynthesize, like what is the stoichiomic pathway, how we can approach to this particular stoichiomic pathway. Likewise, these are very important things when it comes to the biosynthesis pathway and secondary metabolism. I hope you have the idea about it, and in next video we will go more depths into this particular topic.